Hi everyone. In this PowerPoint, I'm going to discuss how this week's material for March 24th and March 26th relates to the different sections of your research paper, and then the majority of this PowerPoint will describe the highlights of two papers that were published back-to-back in Science that were successful in cloning the cystic fibrosis gene, which is the CFTR gene, which stands for cystic fibrosis transmembrane receptor. At the bottom of the slide, I just have a reminder of two upcoming assignments for the week of March 31st. Okay, so where are we in our research paper? So just as a reminder, in the first part, the literature review, this can be up to 8 pages long, and you can decide how long each of the individual 5 sections will be. But in total, this Part 1, composing of the sections 1 through 5, should not be longer than 8 pages. Okay, and then a reminder that the first draft you're working on is for sections 1 through 3, and that's due Tuesday, March 31st. And then the second draft you'll do is for sections 4 and 5, and that's due April 9th, and that is currently posted. For that second draft for sections 4 and 5, you will need primary research articles. So as soon as you can, you'll want to just find which articles will be appropriate to use as sources and try to obtain those papers, especially if you need to get them through interlibrary loan. Okay, so in section 4 of the paper, if we kind of zoom in on exactly what that part entails, this is the section where you are going to be describing how the causative gene for your disease was identified. So this will include first how the gene was mapped to its location on a chromosome, or how mapping was done that linked the gene with the disease. So you'll want to include specific methods. This might be pedigrees and linkage analysis like we see in the Watkins paper, or it could be other relevant techniques. And then after describing the method, you'll want to describe the results. And remember, that's the actual data shown in a figure. And lastly, you'll interpret those results, as the authors did, for at least one of these experiments. So after describing in full a mapping experiment, you will move on in Part B to describe how the gene is cloned and what is the gene. So again, you'll describe methods, results, and the interpretation of results for at least one critical cloning experiment. This nearly always involves screening a cDNA library because you want to describe the actual DNA sequence for your gene. So that will include the size of the cDNA and the number of amino acids of the protein encoding by the gene, and any protein domains that the authors are able to see using software programs. And then lastly, you should discuss how the identity of the gene was confirmed to be a causative gene for the disease. So you need to, in your paper, to describe at least 2 kinds of experiments. Again, describing the methods and results for each. And there are 3 different types of experiments that are laid out in the assignment. We'll go over those today as well. For each of those, you'll describe an overview of the method and the results and interpretation as well. Okay, so now that we've talked about just kind of where we are in the research paper and how today's PowerPoint notes relate to the research paper, which again is mainly section 4, I'm going to describe for you some of the highlights of these 2 papers. I'm going to focus more on the Rordan paper, and both of these papers are posted in Blackboard under course materials, so if you want to refer back to the actual papers, you can. Okay, so the first of the 2 papers, again published back-to-back, so basically at the same time in the same issue of the journal, is called Ramans et al. And the background information they include in their introduction is to remind us that cystic fibrosis is the most common severe autosomal recessive disorder in the Caucasian population. The clinical signs include pulmonary disease, pancreatic exocrine insufficiency, and an increase in sweat electrolytes. But it's not yet known at the time of this article, which was 1989, if the abnormal chloride channel function is the primary lesion or the primary problem in cystic fibrosis patients. An interesting side note is that a long time ago, cystic fibrosis was actually diagnosed by licking the foreheads of newborn babies. Because of the problem with the increased sweat electrolytes, they would actually be very salty, and so physicians could actually fairly accurately diagnose cystic fibrosis by this very crude method of just licking foreheads of newborns. Of course, we have more sophisticated methods these days, which we'll be talking about in future lectures. Okay, so what else did Ramans know before they started their study? They knew from various linkage analyses, these mapping experiments like we saw from Watkins, that the cystic fibrosis gene or locus had been assigned to the long arm of chromosome 7, and on the next slide I'll show you a picture of that. And they knew specifically some of the markers flanking the cystic fibrosis locus or gene were MET and the D7S8 locus. So the goal in this paper was to isolate the cystic fibrosis gene from around 500,000 base pairs on chromosome 7. This is just a picture of chromosome 7, just to kind of remind you that the short arm is the P and long arm is Q. So when we read about in the Watkins paper about those Q positions, that's the long arm, and you can see where cystic fibrosis is located, kind of in the middle of that long arm. In the Ramans paper, they use a method called chromosome walking. So first they made a genomic DNA library, and this was a method they were able to use to clone sections of DNA and then search for candidate genes in these large regions. We're not going to go into the details of exactly how you do this, but it's basically a series of southern blots, a lot of hybridizations, using sort of sequential probes to figure out how these different chunks of DNA line up together, and basically trying to get, the structure of that section of the chromosome. Once you have those pieces of DNA in the right order, you can then test to see if they meet the criteria you would expect for the cystic fibrosis gene. One aspect that's helpful in doing the alignment of those pieces of DNA is that there's synteny for or between related organisms. So this is something you may see in some of the papers you read for your disease. It's very interesting if you line up the different sequences and genes and gene markers between the human chromosome 7 and the mouse chromosome 7. You can see I've highlighted just a few of them. In green, you can see the MET marker that we read about in Watkins, and then you can see this other one, ST7, and then lastly in the lower green box is the CFTR gene itself. So notice the order of these markers is conserved, and you can look beyond this and see that the order of many of these markers is conserved between the two organisms. So that's what synteny is. Okay, so in the Ramans paper, as I said, they did a whole lot of things called chromosome walking and jumping, but what we're going to focus on is how they actually tested some different regions. So you can see in this kind of complicated looking figure that this is the 500,000 base pairs of chromosome 7 that they were testing. You can see the MET marker at the very top, I've circled in purple. That's the same marker from the Watkins paper. And what I'm going to describe briefly to you is how they tested 4 different regions that seem to be potential candidate regions for where the cystic fibrosis gene might be. So we can just call these regions 1, 2, 3, and 4, and they correspond to those red circles. And the Roman numerals that you'll see towards the bottom of this figure correspond to the exons of the CFTR gene. So of course they didn't know where the CFTR gene was when they were searching for these candidates. Keep that in mind. So this is sort of overlaying what they learned later with where they were searching. So what you can see is that actually the first exon is within the 4th red circle. So it was in region 4. And okay, so that was I've just sort of summarized fairly briefly a lot of work that was done in the Ramans paper, and the Rorden paper that is published, again, back to back with that paper, summarizes the work of Ramans, reminds us that the basic defect in cystic fibrosis patients is decreased chloride ion conductance in epithelial cells. And they remind us that of the different candidate regions in chromosome 7, that there is one that meets all the general criteria expected for a cystic fibrosis gene. So the major goal of the Rorden paper is to clone the CF gene and obtain the complete sequence by screening cDNA libraries. So in the first figure of Rorden, they show us this fairly complicated looking figure of overlapping cDNA clones. So what they did here is they screened 7 different cDNA libraries using H16 as a probe. So H16 I've written down in red at the bottom of the slide. That was one of the probes that they had previously identified. And using that probe, they were able to get one cDNA clone from a sweat gland library. So this is the clone 10-1 that I've circled in red at the top of the figure. And it was 920 base pairs. And again, the library that they were using there, a sweat gland library, what that means is it's a whole bunch of cDNAs that are actually made into RNA that are expressed in sweat gland cells. So this was very exciting at the time to finally get a positive hit and to get a clone in the screening after screening 7 different libraries. And so once they had this 10-1 clone, they used this clone to do many more rounds of screening, and ultimately they got 18 more clones from a variety of different cDNA libraries. And they were able to align these different clones and overlap them in order to get a consensus sequence, which I've circled in green at the bottom of the slide. So they did a couple of other molecular methods and procedures, but ultimately they had the complete sequence of the cDNA for this region. It was 6,129 base pairs, which is shown here in figure 2, and this would encode a protein of 1,480 amino acids. Right, so that open reading frame has 1,400 amino acids that would be continuous. The start codon is circled in red. It's very small, but up towards the top left, you see the first methionine. And the stop codon is circled in green. On the top right, it has just asterisks to indicate the stop codon. And as they analyzed the sequence, the software was able to inform them that there were several possible membrane-spanning segments, and they put those in boxes throughout the sequence. And there were also possible ATP binding folds, so they underlined those amino acids. Okay, so by seeing these protein domains, it gave them a little bit of a hint as to where this protein might be located. It might be actually in cell membranes, and that might be consistent with this problem that they had seen with chloride ion conductance. So this is very exciting. Once you have the sequence of the gene, you've cloned the gene, you can now explore the function of that protein in ion transport and ultimately determine what is not going correctly in the patients and how to potentially provide therapy. In the Rorden paper, the next thing they do in figure 3 is to show us some RNA analysis. So on the right-hand side in the box, I'm reminding you or telling you that an RNA blot, which is how they've labeled this figure, is the same thing as a northern blot. So RNA from various tissues, and those are listed across the top of the gel, the RNA was run on a gel, transferred to nitrocellulose, and then probed using that 10-1 cDNA. And so the idea here is to ask whether or not this gene is expressed in each of these different tissues. A control of sorts is shown on the far left. The T84 is a colon carcinoma cell line, and so they see a strong band there. The results from their tissue analysis were strong bands in the pancreas and nasal polyp lanes. Those are on the left. They also saw bands in lung, colon, sweat glands, placenta, liver, and parotid gland, though some of those bands were pretty weak. They did not see bands in brain, adrenal gland, or testis. And although not shown on this figure, in the text they tell us that a band for kidney was eventually seen on a later northern blot. Okay, so this northern blot, I should just mention as a side comment, is kind of dirty. It's not just clean bands. You can see a lot of sort of faint bands below the strong band, which is the 6.5 kilobase transcript. So the arrow on the right-hand side of the gel is showing us the size of the expected RNA. And the other kind of faint bands we see are probably not significant and not going to affect our interpretation of what's going on here. Okay, so this is exciting. They've cloned the gene, they have the sequence, they see that this gene is expressed in some of the tissues that they would expect, right? The tissues where CF patients have problems. And so the next thing they need to do is confirm that this gene really is the correct gene for cystic fibrosis. Now, we've talked about this in Module 2 several times. How can you confirm that a candidate gene is correct? You can find mutations in the DNA sequence, and finding patient-specific mutations is really the gold standard most of the time for proof of principle that this is the correct gene that's causing a problem in the patients. The two other types of criteria would be to kind of rescue, which would be to restore the normal phenotype, and that could be in vitro using cell culture or an animal model where you could transfect or otherwise transfer in the normal allele to rescue the mutant phenotype and actually then see a wild type phenotype. Gene therapy would actually be another method that would be a rescue type method where you would be providing the wild type copy to the patient, hoping to restore normal function. The third type of criteria would be to actually phenocopy. So we talked about using RNAi to do this in Module 2. You can also make a mouse model of the disease. So you could make a transgenic knockout mouse, and if you knock out this gene, you would expect that it would mimic or resemble the phenotypes of the human disease. So in the Rorden paper, they only do the first type of method. So they're going to show us in Figure 4 the DNA sequencing from normal individual compared to a cystic fibrosis individual. So in Figure 4, they're showing a sequence from nucleotides 1,627 to 1,651. And this is a real old school sequencing gel. So in this kind of figure, what you're seeing is that each lane is a nucleotide. So I've actually labeled those in blue at the bottom. So if there's a band in the far left lane for T16, that would be a G. In the second lane, it's an A. If there's a band in the third lane, it's a T. And if there's a band in the fourth lane, it's a C. So you can see how they've interpreted the bands on the left for left of normal or right of CF. They actually are telling you what the nucleotides are. And if you compare, what you'll see is where the blue arrow on the right is pointing that there are 3 nucleotides, CTT, that are missing in the CF patient. This corresponds to one phenylalanine amino acid at position 508. So this mutation is called delta F508. And this is the mutation that is seen in approximately 70% of cystic fibrosis patients. So this is exciting. It helps confirm that they have cloned the correct gene for CF. It's also exciting because now that they've got the sequence and now that they see the mutation in the patients, they can try to figure out why this particular mutation causes a problem in the function of the CF protein and ultimately figure out the pathophysiology, right? Why is missing this one phenylalanine resulting in all of the symptoms and phenotypes that we see in cystic fibrosis patients? So stay tuned and we'll learn the answers to that at a later PowerPoint.